This is TWIS. This Week in Science, episode number 821, recorded on Wednesday, April 21st, 2021. Putting science in the light. Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your head with tarantulas, beetle pee, and a bit of light. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. Nothing is in the way of the world doing all the right things to prevent climate emergency from taking over. That looming, dooming, glooming over our planetary future like a dark foreshadowing cloud of something dystopian, ready to burst with thunderous metaphors, the sort of mechanisms used in literature to alert readers of the dangers to come. That the story, in this case, isn't fiction is a little bit of a problem. Instead of literary props that hint at the action ahead, we have scientific papers that spell out the threats, the causes, the solutions, consequences, and outcomes in great detail. If you watch the news these days, you won't hear anything about it. That's because there's no story arc, there's no reveal, there's no ratings appeal. It's just spoiler alert after spoiler alert. Like a whodunit, where the real killer is revealed on the first page. A mystery twist movie where the big secret is revealed in the trailer and the title gives away the inevitable end. And so, with nothing left to the imagination, nothing left to interest or entertain, the public simply switches the channel, tunes out the emergency warnings. Or they might do that if the emergency warnings were ever aired in the first place, but they aren't. You won't see it covered anywhere. While we seem to be saving the end of the world for some sort of late century news cycle, we here will continue to bring you the boring news about the planet you are on, the universe you are in, and the uneventful discoveries about how everything works. Here on This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about discovery, curiosity, findings, lots of findings, things of interest to our brains. And we do hope that you enjoy the hour plus that we have in store for you today. What did we bring to the show? Well, I have some stories about, what did I bring? I've got news stories about chimeras, honey, T-Rexes who are kind of slow, and we have an interview tonight with Gbur. Yes, looking forward to that. I'll be introducing him in a bit. Hello, Justin. Yeah, what did you bring to the show, Justin?
I've got why humans aren't all bad, traveling tarantulas, and is shift work really bad for you? Spoiler, yes, it is.
Spoiler, we've talked about this before. Blair, what is in the animal corner?
Heyo, I have coffee, dogs, beetle pee, snake venom. Bam.
Wow.
Bam! Choom, choom, choom. Well, we are going to get those stories out there for you all. But before we jump into the science, I want to remind you that if you have not yet subscribed to This Week in Science, you can find us at twist.org. You can also find us on YouTube, Facebook, Twitch, and every podcast platform that's out there. Look for This Week in Science. TWIS. All right, everyone. Let's dive into some fun. Chimeras. I would be missing a step if we didn't talk about monkey-human chimeras today. I know that we talked about them a little bit over the weekend in our special crossover show with DTNS, which was loads of fun.
Yeah, it was
and
a blast.
that podcast episode is going to be released to patrons only. So if you're not a patron, like that's a place where you can listen to the podcast episode. However, chimeras. When you think of chimeras, maybe it takes you to mythology and the idea of these beasts that are half lion, half man, half bull, a variety of animals that are mixed up together.
Is that a thing? A griffin?
A griffin?
Something along this line.
Yes.
Is it one of those type of
exactly
creatures?
An amalgam of creatures that work together as a creature with the abilities of these various creatures that somehow makes them stronger. All right, so here is the bottom line for research, scientific research that is looking into putting human cells with the cells of other species. The idea is that we could one day be able to produce organs for human transplantation that would be acceptable to the human body, not rejected by the human body as transplants from other species are now. We have to use lots of anti-rejection drugs. And that we could have a source for organs for people. And we really need a source for organs for transplantation. That is a dire need for the health of humanity. So we're working on this. So far, human-pig chimeras haven't worked out really well. About 1 in 100,000 pig cells is human when we try and put human cells into pigs. It doesn't work out very well. And so we've tried it now. A group in China went through a lot of review, a lot of ethical review, bioethicists taking a look at the study to see how they were going to produce it. In China, there's another researcher from the United States who was involved in this, Ispazua Belmonte, and he worked with this team in China to achieve this goal of a human-monkey hybrid. Now, this is not a full-grown animal. This is a clump of cells. It's a blastocyst that was allowed to exist for less than 20 days. Not allowed to come to term. This is a ball of cells. There is nothing to this. It's not even an animal.
And but so that's also so far, isn't it?
Like, so far. And at this point, they don't have a plan at this point to allow anything to move past the ball of cells stage. At this point, it is looking to see if they can do it and what happens. Some pretty neat stuff, though. They can actually see this combination of human and monkey cells as the chimera, this embryonic ball of cells, grows. They marked all of the human cells with a red fluorescing protein. And in that way, they were able to track the cells to be able to watch them as they integrated into the blastocyst to see how they integrated. In the process of the blastocyst growing, they discovered that the human cells didn't really like to hang out with the monkey cells. And they were like, I'm going to be over here. You go do your own thing. And the human cells that did actually make it inside this embryo, they kind of hung out with their own kind of cells. And so there are obviously some issues with integration that the researchers don't understand yet. And to that issue on top of it, there were a number of places within this human monkey embryo in which the human cells just didn't take at all. And so this is not exactly a successful experiment in terms of the end goal of creating a human monkey hybrid that can create organs. This was not successful, but it did give information, and that aspect of it was successful.
You know what this reminds me of is cat chimeras, which are completely different, of course. But you're telling me about how the human genes and the monkey genes kind of stayed apart from each other, but that's it.
Not the genes, but the tissue.
Or the tissue.
The little cells in the ball of cells were like, I'm going to be, this is the human area, and the monkey ones are going to be over here.
Right. And so when there are two embryos that fuse, they end up with this very stark difference in the way that they look. And so it could be similar that as it's developing, these two genomes are like, I'm different. Keep me apart.
So I would take it away from the genome aspect of it, though, and the reason why is because the genome is more ingredients, sort of, right? Okay, and you get then the tissue that forms based on this. And what's interesting is that the tissues themselves have a level of communication to each other that genes don't talk like this. But tissues do. Tissues are in communication. Tissues are relying on their neighbor and the signaling from their neighbor and how that neighbor is signaling. So it's sort of interesting that even when you get these genes combobulated together where you're trying to create the hybrid activity, when it gets to the tissue level, they don't speak the same language. That to me is sort of fascinating.
Yeah, and another aspect to this is that they looked at the transcription factors, the proteins that were the genes that were being transcribed, and what was getting turned on in the process, and they found that in the human monkey hybrid cells, in those areas, these balls of cells did not differentiate and didn't develop at the same rate as either a only human or only monkey blastocyst would be developing. It slowed down the development by putting them together. And so there is miscommunication. They found there were different pathways turned on. There were unexpected proteins and genes to be activated. So there's a lot to be learned here. And they're maybe looking at potentially which pathways do get turned on and how those could be important moving forward in this technology. Well, anyway, there is a whole lot of work that still needs to be done for human-monkey hybrids. This is not a portent of the ape man to come in the future. There are a lot of issues that still need to be worked on. And it does sound as though they are really doing their best from a scientific perspective to take ethical, moral issues into consideration in the work that's being done.
Well,
that said, it was done in China because no other countries would allow it to be done.
But you also have to understand what the first and second choice options were. The first option was just harvest organs from poorer humans in third world countries. That was option number one. That's where we were headed.
No, that's not good.
That was the first option. And so people were like, what if we clone ourselves and then harvest the organs? And then they realized, we're going to clone humans and then steal their organs. That's almost as bad as the first one, if not. And then the third option was like, we'll make like an analog hybrid that we can harvest the organs from. People are like, okay, that's not nearly as horrible as the first two options.
Yeah, so, and I think that's this brings up a really good point, and perhaps a future conversation for future crossover episodes.
That Justin watches too much dystopian sci-fi.
Yes.
Maybe that's the issue.
But just that if you're gonna do the preliminary testing to see if this is possible and viable, that means at some point you want to grow a full chimera. Mm-hmm. So the ethics conversations have to start now, because it's going to take a long time to figure out the kind of rules of engagement for this future realm of study.
Yep.
And if you think I'm talking about dystopian future, Google India kidney black market. Just
I think I don't want that in my search history.
It's already a thing that's been going on. It's not. Okay. It's not a distant future scary thing. It's a now, yesterday, and for decades, actually, thing.
Oh, okay. But so you're saying that, and that makes me go, humans are bad, okay? But Justin, can you tell me how we're not so bad?
Well, they're not. So the invasive species that is currently destroying the world's air, water, soil, disrupting fauna, foliage, selling each other's kidneys to each other, pretty much putting plastic into every earthly orifice, isn't all bad, or at least wasn't always all bad. Human recklessness and resource harvesting is relatively recent. kind of a new thing, according to new research published in the Proceedings of the National Academy of Sciences, PNAS. Human societies for the last 12,000 years have been reshaping the ecology across most of the Earth's land surfaces. We sort of tend to think of this as a new thing, that we've gained control of everything on the planet. But according to them, actually, indigenous people have spanned the earth and have been farming and hunting and managing the wildlife around them for more than 10,000 years. The difference seems to be a more modern approach, is where you got the sort of resource, the clear cutting of every resource imaginable. But yeah, they look back over 12,000 years. 3/4 of the terrestrial nature was inhabited, used, and shaped by people, says one of the researchers. Areas untouched by people were almost as rare 12,000 years ago as they are today. So granted, there's a lot more of us. We're doing a lot more transformation. But there was an interesting statistic that I thought was in here. This is from somebody from the Penobscot, can't pronounce it, Indian nation, notes that indigenous people currently exercise some level of management of about 5% of the world's land, which is sort of a little off-putting if you understand how much it was just saying that the people had been managing the majority of the terrestrial areas on the planet. And now the indigenous people, those people who've been apparently managing that land for a while, are now only managing about 5% of the world's lands. But in that 5%, 80% of the world's biodiversity exists. So while indigenous people have been excluded from management, access, and habitation of protected lands in places such as the U.S. national parks, the track record says maybe we should let them have a larger voice in how things are operating. But yeah, humans, not always bad for the environment.
We haven't always been, and maybe we can relearn some of the lessons that we've forgotten or that we pushed away because we were so bent on.
Well, and you know, here's the sort of interesting thing: like
land.
If you combine it with the indigenous thing, then it makes sense. It becomes very NIMBY all of a sudden, right? Like if you are living in a beautiful forest, you're not. Hey, you know how we can make money? Let's level our home. Let's knock it down. You know, I bet that I bet if you're in a really nice big house, you don't go, you know what? I bet there's plenty of copper in these walls. All the wiring and the lighting we rip out all the copper. We could cash in on it. Nobody does that because they want to live in their home. And so I think that the point of that story also is that if you live where those resources are, they're not just resources to exploit. They're part of your home. They're part of the infrastructure of your home. And I think that's what happens when people have power over land in which they do not inhabit. Is that they can see it separate from being home or habitat.
Yeah, that it's then a combination of the two. 12,000 years of history, huh? We can get back there. We can do it. But Blair, maybe I'm forgetting things a bit. There, maybe some coffee could help me remember.
You want to talk about coffee? I love coffee. We should talk about coffee. I need coffee.
We should talk about coffee right now, okay? Yeah. No, I have not had any. I can't speed up.
You see, coffee is really it's impacted by climate change because coffee, those tropical plant, it's really temperature sensitive. So it can't get too hot, or the coffee, you'll lose all the coffee. And then what will you do in the morning? So
just cry. Just cry.
I was actually heartened by this recent study where they actually it was less of a study, more of a discovery. They discovered a type of coffee bean that was thought to be gone. And I didn't know this. There are over 100 known coffee species. I guess I just really hadn't looked into it. The world gets all of our caffeine from just two. Arabica, which is the superior brew, that's usually what you're drinking, and Robusta, which is usually used for instant coffee. So that's why instant coffee is so gross. It's a different bean. This is what I've learned. And
Wait a sec. It's an acquired taste.
It's bad. And even these coffee researchers say it's bad. It's gross. So the issue here is that in the multi-billion-dollar coffee industry, over 100 million farmers are earning a living doing coffee. And so the problem is that Arabica is from the highlands of Ethiopia and South Sudan. It's a cool tropical plant. Its average annual temperatures are around 19 degrees Celsius. And it is really sensitive to temperature change. Any change past that 19, and it starts to not do so well. But Robusta actually is more robust, you might say, and can endure up to 23 degrees Celsius. So this other coffee, Stenophylla, can tolerate conditions similar to Robusta, but it tastes good too.
Ooh.
So here's the thing: it's more resilient and it has good taste. Now they thought it was gone in about 1954. It completely disappeared, Stenophylla, from studies, from records, and so it didn't get used because they thought it was gone forever. But they found it growing wild in Sierra Leone in 2018, and then they started testing its temperature tolerance and its flavor. They did blind taste tests. They found that this coffee was good, and they do think that it could become commercially available, but it would take some years because they found this vulnerable plant. It's listed as vulnerable on the IUCN Red List. And so they would have to cultivate this, kind of tame it to be farmed a little bit, find the right place to grow it, create an industry for it. But they think in some short years, this thing could pop up in your coffee cup in the morning, and it might be a lot more resilient to climate change.
That's
Boy,
what I was.
They should have kept it a secret.
Yeah.
Because now you're going to have that. You know what's going to happen. These sort of baristas, coffee snobs are like, I've got to get my hands on that bean. I got to just try a cup. And they all try a cup, and then it's all gone. And nobody can have any because
this is the beauty. That actually puts money into the farming of it and
make it more available,
which actually makes it more available and the population more robust as well. So.
Yeah, I love the idea because we've been talking so many times on the show about the potential plight of coffee and chocolate. Two very important compounds that we really enjoy in our culture. And if through climate change, it gets harder to be able to grow those beans in the places where they're known to be grown, you need a more robust plant. So why is this plant vulnerable if it is robust? This is...
So
what happened to it?
So it's more robust to climate change. That does not mean that it's more robust to invasive species, poaching, all these other kind of, habitat destruction that Justin was just talking about. So it's still a rainforest species, and it's still therefore in a lot of trouble. We got to save it and cultivate it.
Save the coffee. Save all the coffee. We need it. I think we've got some more stories, but let's jump into our interview. I'm going to dive us forward a little bit in time to now, everyone. You're watching This Week in Science. And if you like the show, please tell a friend. We'd love it if you helped spread the word. I would love to introduce our guest for the evening. Dr. Greg Gbur is a professor in the Department of Physics and Optical Science at the University of North Carolina, Charlotte, where he studies classical optics and the wave nature of light. He's also the author of Falling Felines and Fundamental Physics, which I fully approve of. Welcome to the show, Dr. Gbur.
Oh, thank you. Great to be here. I've just been enjoying listening to your early discussion.
Yeah, we enjoy talking about all the things. And your visit to the show tonight came about because of something we were talking about on the show last week related to laser beams and scattering light and the possibility of light making its way through objects, through opaque surfaces, which is a fascinating idea. And someone shared a post of that with you, and you're like, wait, what? You could talk to me. And so we're talking to you now. I'd love to get your comments on this story. But I'm also so excited to get to talk with you on the show because you were on my old show, Dr. Kiki's Science Hour, years ago when I was not on my show because I was in maternity leave.
Yep.
Oh, wow.
Yep, I remember that. I didn't know if you'd remember that.
Yeah. So you had a wonderful conversation with Brian Mallow, and I wasn't there, and I was sad. And now I get to talk with you, so I'm very excited. And we all get to talk with you, so it's a gift to everyone. Yay! Just to start off, we're not going to dive right into that story because I want to bring people into who you are and what you do and how we can how you can help us understand what we were talking about last week. How did you get interested in optical physics, in light? Like, why the waviness of light particles as opposed to the particle aspect of light particles?
Well, part of it is my history as a confused graduate student. I originally went to graduate school to do high energy physics because I thought I wanted to study the secrets, the fundamental secrets of the universe. I still do technically, but there's plenty more to study. And so I was doing a lot of work on experimental high energy physics, but started to feel like it wasn't for me. I wrote a blog post about this years ago. There were like a handful of reasons. It wasn't like there was one particular reason. It's a great field, but there was just like a bunch of little stuff that made me think, maybe I don't want to do this with my whole life. A friend of mine was working for Professor Emil Wolf, who is one of the big names in optical science and theoretical optical science. And my friend said, hey, why don't you go talk to him? And so I talked to Professor Wolf, and he dumped this pile of papers on my lap that he had written over the past 3 or 4 years. And I really fell in love with the elegance of the wave nature of light, the mathematics of the wave nature. If you look at pictures of waves, everything looks really beautiful and sort of elegant and smooth. And that's really how the mathematics feels too. It really makes sense when you look at all the pieces, they fit together perfectly. The quantum nature of light, the idea of light as this quantum particle, we're still actually trying to figure that out. I don't think anybody really completely understands. We can predict experiments great, we can do experiments great, but trying to understand what it all means is still kind of a big question mark. So I love the elegant mathematics, which is what drew me in, and I'm still trying to personally figure out all that quantum stuff.
This could be some deep quantum soul searching on the personal side. Yeah. So what is it about optical physics that you work in that just you find really interesting? Like, what is this like? What just drives you? What do you want to figure out?
A lot of what I like to do now is the way I describe optics these days is if you look at the history of optics over the past literally 1,000 years, it was all about understanding what light can do. Like, okay, light can go through this small hole, it can't do that, and just sort of setting down the rules. Over the past kind of 20 years or so, the story has changed, and people have realized that a lot of these rules we have taken for granted aren't really rigorous rules, and they can be bent. And so the story has become more, instead of what can light do, the story has become how can we make light do whatever we want it to do.
Yeah,
and
what can't light do?
Yeah, and so that's a lot of what appeals to me is seeing all these different ways we can break the rules. I mentioned like imaging. The wave nature of light means that that when an object is too small, you can't really see it anymore or get a picture of it with ordinary light. But people have figured out that by doing a variety of different tricks, you can see things smaller than the conventional wisdom would tell you could get away with. And it's really fun to play with. It's both the fundamental part and the practical part, because you're trying to get around seemingly fundamental rules, but then you can actually do something with it in the end, if that makes some sense.
Yeah. So When I think of imaging, it's a lot of light bouncing off of stuff. And then we are recording or viewing that reflected light, right? And so with X-rays, it's a bit different. That's just more energetic light when you start thinking about it. But how do you break down like the various wavelengths and what they can do and what they can't do? How do you start looking at that?
Well, the different wavelengths, it's really, in the end, it all comes down to how they interact with matter. That's what makes the difference. So yeah, X-rays are really high energy photons, we would say, light particles, and they just barrel through stuff. Some of them get absorbed. That's what allows us to take X-ray photos. And it makes the math really simple because you just shine X-rays in one side, and on the other side, you just measure how many got through, and they all came through in a straight line. You can deduce kind of roughly what they went through. At other wavelengths, different things happen. If you're looking at infrared light, that tends to excite thermal vibrations in molecules and atoms. So that's what makes things heat up. And ultraviolet light tends to kick electrons off of items, which is part of why it causes cancer, and does a lot of chemical reactions. So breaking it down, the behavior of the light really comes from our matter-based perspective, that we're always looking at how these different wavelengths interact with matter.
And that gets at an area that you're interested in, which is the inverse problem. And that is looking. Can you explain the inverse problem? Because I know if I try to explain it, I'm going to completely hack it to bits. And so, if you can...
Sure. Yeah, I'm actually trying to write an explanation that makes sense myself these days.
Yeah.
So I think I've been thinking about it. Well, one way to think about it is most problems in physics are sort of they follow sort of a cause and effect scenario. So you say, here's an object. I know what this object is. I hit it with a hammer. I want to predict what happens. I'm going to use the laws of physics to figure out what the effect is. If I use a hammer as my cause to whack an object, I can use the laws of physics to figure out what the effect is on the object. The inverse problem is going the other way around, saying, suppose I measured what happened to this object that I smashed. Can I now go back and figure out either what the object was in the first place, or can I deduce what the cause was, what I hit it with? I often describe it like a crime scene investigation, that at a crime scene, you show up, you want to know who did it, and you want to know what was done. You've got a certain amount of information. You're trying to backtrack and figure out who did it, what the cause was of the phenomenon. So an inverse problem is reversing the cause and effect rules that we usually use in physics. We go from an effect to a cause.
Isn't this how we talk about the Big Bang?
To some extent, yeah. I mean, in a sense, that's very much a sort of inverse problem, that we've got the existing universe as it is. We kind of know the laws of physics, so we're kind of backtracking and trying to figure out where it all came from. And we talked about X-ray imaging. That's another example of an inverse problem. Or CAT scans you can get. You take a lot of measurements. You don't know what the object is you're imaging. You measure what happens to the X-rays. And then from knowledge of what happened to the X-rays, you try and figure out what the object is.
And so when we're coming from a matter-based perspective, is there anything that we are aware of that might be there, but we're not perceiving it because we don't know how to measure to get the cause?
Well, to some extent,
like invisible objects that are there, but
well, I'm not. Yeah, that happens to be an area that I study, is sort of invisibility theory, and I don't know that there's anything there we're not seeing, but this is actually a problem in inverse problems. This is why there's a whole mathematical field called inverse problems, because pretty much anytime you're using your eyes, you're also doing an inverse problem. You look at something, your eyes detect the light, you deduce what that object is you're looking at. But those are really simple problems that you don't have to worry too much about hidden things, though there are tons of optical illusions that show that there are a lot of problems with the way our eyes detect things.
Yeah.
But you start doing like computed tomography, where you take a bunch of X-ray images of a person from different angles, you take all that data and you mix it up in a computer and get out an image. And now you run into the question, okay, I did all this math, but how do I know that I'm actually seeing everything that's there? And this ends up being something that's not talked about a whole lot, but there's this intimate connection between invisibility and inverse problems, because in the context of a particular problem, if an invisible object exists, if invisible objects in principle exist, then you can't solve the inverse problem, because whatever you measure, there's always the possibility that you're missing something. And I use the crime scene investigation argument for that too, because there is the risk when you're studying a crime scene that there just isn't enough information present at the crime scene to solve the problem. And that's also where invisibility and inverse problems come together, is you really worry about when you're studying a new type of inverse problem, whether there's enough information there to figure out what you're trying to look at.
And so is that just a matter of us needing to catch up with the unknowns that might still be there? Sort of, I'm picturing this crime scene happening a really long time ago, where this crushed body is found at the bottom of a cliff face, but they haven't figured out gravity yet. So they're like, something big must have stepped. That's the only solution, because they're very squished. But
well, one thing, to use a technical term, all the technical terms in physics are often very boring. They use what's called prior knowledge, because sometimes you don't have enough information on the scene to figure out what happened, but there's extra information you could bring into the picture that helps. So in your case, you find this guy lying crushed on the ground. You say, well, I can't tell if he fell off from a big height or he got stepped on. And then you look over there and there's a big cliff. You go, okay, now I can use that information. All right, that helps me.
Context. Yes.
Yeah. So that's a big trick in a lot of these sort of imaging problems, is taking advantage of information you already have. Like in medical imaging, we know that the patient that we're imaging is roughly human sized. So even the size of the person is an important piece of information. We know we're not imaging somebody the size of a house. So when you get that data, you have some context. If you get a solution that says this person is the size of an elephant, you know that's not the right solution.
Right. It's good to have those limitations on you that set the boundaries that can contain the information that you'll find that will be relevant to what you're looking for. So from the invisibility standpoint, how close are we to invisibility cloaks? And I mean, is Harry Potter around the corner, or is this like going to be like, super secret CIA tech?
This is one of those dangerous questions to answer, because I'm on camera being recorded about it.
I know.
And so it's fair. I could give my conservative estimate is any time from tomorrow to 100 years from now to never.
That's a super meaningful answer.
Range.
Thanks. No.
Well, to give a little context.
So not in the past.
No, it definitely not in the past.
That narrows it down quite a bit. There's a lot of past. That's billions of years we ruled out right there.
Yeah. To give a little context, so in 2006, the first papers came out about the theoretical possibility of invisibility cloaks. And I had done my PhD work on some invisibility stuff. So national newspapers contacted me and said, what do you think about this? And they naturally asked me, when do you think something will happen? And I was like, well, you say 5 years and nobody's going to remember what you say in 5 years. Yeah. Well, it turned out it was more like 4 months that they made a flat invisibility cloak. So it was just this small thing. They were sending waves through this small slab, and it was designed for microwaves, and it was not perfect, but they surprised me by already knowing how to demonstrate the principle. But to give a better answer to your question, it's kind of hard to, it still is kind of hard to say. There are a lot of really amazing advances that keep happening. So one big problem with invisibility cloaks is that, so the idea of an invisibility cloak is the light comes in, it goes around some hidden region and goes on the way it was. But the catch is that the amount of time it takes for the light to take that detour has to be the same amount of time it takes for light to go on the outside of the cloak. Otherwise, you could detect that time difference and you could see that something was there. That means that the light going through that cloak is probably going to have to go faster than the vacuum speed of light. And we know that it can't do that. Or, well, it starts to get technical, but it can kind of do that for like one frequency of light or one wavelength or a very small range of wavelengths. But so you can make an invisibility cloak that would work for a really particular shade of red, which may be useful. But so that's been a limitation. But last year, somebody else, I was talking to another podcast person about invisibility stuff, and they're like, "Oh, did you see this paper?" And I was like, "What paper?" And some researchers kind of found a clever way to partially get around that limitation of this will only work for one color. So the biggest problem is still materials. In order to make an invisibility cloak that guides light that way, you need to make a device. You have to be able to control the structure of matter in three-dimensional space. You have to make a three-dimensional object and kind of be able to control the structure of the matter almost on the atomic level. And
this doesn't sound like something that you'll be able to just throw over your head and walk around with. This
also sounds like the hard way to do it. Why isn't it just, cameras on one side, wearable screen on the other? Why isn't that just like the shortcut version?
That's a very astute question because that has been done. And in 2003, there was a Japanese research group that they made a retroreflective coat, and they had a projector on the front and a camera in the back, so the person looks completely see-through. And it's a really eerie image of them just sort of standing there with their face not visible, sort of waving around like a ghost. What I thought was really cool, but they're not really invisible because you can still tell that they're there. But the really cool thing about this is what you can do with it. And that is, in their case, they're trying to design a transparent cockpit for an airplane or for the interior of a car, so that you're in your car
everywhere.
Yeah, you could see if you're about to hit that car right to your left because you can literally see it. Or if you're flying an airplane, you'll know if there's a plane right below you because you just look down at your feet and you can see it. So that's one of the things that fascinates me about invisibility research is it's not clear if we'll ever make a really practical cloak where somebody can walk around and they'll never be seen. There is the possibility that this sort of technology of making things at least virtually see-through will have a lot of advantages.
I can imagine the advantages for entertainment. I can imagine advantages. I'm sure there are political or mostly I'm going in the media direction, but maybe that's because I spend a lot of time in the media. But from the other perspective of how things like this work, I mean, you have light not really going through things, but this is what that paper was about last week, creating.
Wait a sec, not really light. Going through things? Wait, I thought it was going through things. You're ruining it.
I know. I'm sorry. It's just difficult. Yeah. So last week, the story was about a class of light called scattering invariant light that the researchers showed they could get to pass through a zinc oxide coating, a zinc oxide, quote unquote, object. What do you think about this work?
It's actually really cool. And part of the reason that I was like complaining on Twitter, like, you should have me on, is because I'm familiar with the earlier work of these researchers. They've been doing similar work since like about 2007. And the idea is that, yeah, you've got some sort of material it's strongly scattering but not absorbing. So the light doesn't get like sucked up by the material. It just bounces around in it like a ball in a pinball machine. And so any light that comes out the other side is basically randomized by the time it comes out. This is the difference between like a transparent object and a translucent object. If you have a translucent piece of glass, the light's definitely coming through, but you have no idea what's on the other side of it. But these researchers have been developing this idea that if you structure the light if you pre-structure the light in just the right way, you can start transmitting images through it. And if for a general take, it's just a really cool approach. And it again fits that idea I was saying before of optics is nowadays about doing things we didn't think were possible before. Like if you go back 20 or 30 years, nobody would have said, hey, we can get light through this sort of opaque scattering material. And these folks have shown that by this new technique that, yeah, they can probably even send images through.
How does this happen? What is this? So scattering as a random chaotic process versus scattering as they're calling it, invariant. So obviously there's going to be predictability about how the light is going to move through the surface. Are they predicting this at atomic scale?
No, from what I understand, looking at the paper, they really took a particular material. They basically just measured they sent a bunch of light beams through it to get some idea of how light transfers through the material. They don't care about the structure of the material. They only care about how light gets through it. You send a beam in the upper left corner and you see some of it comes out in the lower right, and then you send it in the upper right and it comes out the lower left, something like that. And you put together this table, basically like a list of saying, if I put light in here, it comes out here. And along the way, they figured out that they could make these so-called invariant modes. And the idea of an invariant mode is they were able to mathematically calculate that if you send a beam of light into the material with just the right patterning, it will go through the material as if it had just gone through air. And yeah,
that is wacky.
So basically, they model the beam specific to the composition of the material while looking at the backside to sort of get the image in focus. So they're cheating. If they moved this like an inch over in the same material, it might not work if the composition is a little different. Yeah,
if you use that. So yeah, I think though if it's the same type of material, I think things will work out on average. But yeah, if you use like a different material, or they were using some sort of material deposited on a piece of glass, I think. If you deposited it in a different way so that the structure was different, then you'd have to restart. Yeah.
Okay. So we're not quite at because I've invested so much money in X-ray glasses over the years. The back of the comic book. I was hoping every year there'd be like a new edition, because the old ones didn't seem to really do the trick. But we're still not there yet.
No.
Okay.
Yeah, this is well, it's kind of like Invisibility. I keep telling people, I'm like, it's really, really cool, and it helps us understand a lot about light, but we're not at a point where we're nowhere near a point where you have to worry about your personal privacy.
Right. You don't have to worry about the invisible person sneaking into your house. And we don't yet have to worry about the laser beam imaging system that's going to see through the walls of our house.
No.
Cover your webcams.
Yeah.
Everybody.
Still, cover your webcams. I think that's
Get your little covers, but other than that, you're fine.
At the nano scale, like when light is interacting with bits of atoms, right? When light is interacting at this nano scale, can you predict what's going to happen?
Yes. Well, yeah. If you have a material that's sort of the what we would call the forward problem, or the cause effect problem in physics. If you know the structure of the material, you can do the math. We understand pretty well how the interaction of light with matter. Though there is a catch, because it's very easy to do the study how light scatters off of a really small object, and it's really easy to figure out how light scatters off of a really big object. When it's an object that's somewhere in between, then things get really tricky. And then we know in principle how to solve that problem, but in practice, we don't have a lot of good tools to do it. And this also goes back to kind of the computed tomography, the X-ray tomography stuff. We know how to use visible light we know how to use X-rays to go through an object. We know how how X-rays travel through an object, so we can work with that. If you try and image the human body, the interior of the human body with visible light, now there's so much more scattering and the problem is so much more complicated. We still don't really have a good handle on that. And as you've probably noticed, we are not transparent, so the problem is quite difficult.
Yeah, and even with cells, just cellular imaging, not even a living person, a whole body, you have to usually kill the cell in the process of imaging it. That the process of imaging itself destroys it in the act. So it's getting inside of biological structures is hard.
Yeah. By coincidence, my former PhD advisor, Emil Wolf, he invented this technique called diffraction tomography, which is sort of it's like computed tomography with X-rays, but it works with visible light and the light is scattering. But it only works when you have really, really small objects. But it so turns out that for people who are interested in imaging cells and getting three-dimensional structural images of cells, it's a great technique. So even recently, there have been a few papers that have come out where people said, oh, look, we can take a living cell and we can actually get some three-dimensional structural information of this cell while it's just sitting there doing its thing.
Amazing. Yeah, we need movies. I want movies that are not visualizations, or, I want the real movie of the actin walking down the myosin.
Right.
We want those, the real thing.
Yep.
But yeah, in our chat room, somebody wants to know, interesting, all this is very interesting, but how many cats does Greg have?
Technically, I have 5 of my own, and my roommate has one, so there's 6 cats floating around at home.
And are they actually floating? Have you managed?
They're part of some sort of experiment you've been working on.
No, I've figured out that over the past 300 years, there's been enough research on cats falling all over the place. So, no, they're just pretty much sitting around. They're cats. They're mostly just laying around, except when I'm trying to do an important Zoom call, and then they're
pay attention to me.
Yeah, then they're sticking their tails or whatever in my face.
But in your book, Falling Felines and Fundamental Physics, you took inspiration from your cats.
Yeah.
How did that? What were you just watching your cats and you're like, how do they always land on their feet? And now I am writing a physics book.
Well, the original thing that got me started on the book was my blogging, because I love to read and write about the history of science. And being a really super big nerd, I tend to get bored, and late at night I'll just go browse old scientific journals. So I'll say, all right, let me start in the year 1850. I'll pick a journal and just go look through and see what interesting scientific papers have been written. And honestly, I worry that even my scientific colleagues find me boring at times, because before the pandemic, I was going to meetings like, let me tell you this little cool bit of trivia. And they're like, yeah, it's a bedtime, I think. But so I found this paper from 1894, which was the first high-speed photography of a cat flipping and landing on its feet.
Oh, fantastic.
And it turned out it caused quite a ruckus, caused quite chaos in the scientific community for a while. But so the first thing I did is I wrote a blog post on that original paper. I'm like, oh, this is cool. It's history of science, physics, and cats, 3 things that I love. And then I got curious. So I was like, is that the whole story? And so I kept doing literature searches, and I just kept finding more and more papers of people studying how cats land on their feet in different fields of different scientific fields, going from 300 years ago up to the present day. People scientists constantly wondering how cats do it. And at that point, I was originally talking with my publisher about writing a book on the history of invisibility physics. And I contacted him and said, I think I'm going to write a cat book first. And they were just sort of like, okay, whatever. The quote in the original 1894 translated paper from Nature says that the expression on the cat's face in the photographs indicates a definite lack of interest in the science of it. Paraphrasing a little bit.
It's so interesting too, because so many animals, I can tell you from personal experience, do not land on their feet.
Right. Yeah, if you can look up some very sad images of people that people did back in the day of dropping dogs that are trying to land on their feet, and the dog is like paddling in the air, desperately trying to turn over. And it's always done with cushioning. And nowadays, I tell people there are enough videos online. Please don't drop your animals. Just go look it up.
You don't need to do these experiments anymore. That's okay. We've done them.
And now that I've written this book, it's kind of amazing that I see this motion in other animals. Just last week, somebody on Twitter shared an image of a mongoose fighting a cobra. And the cobra strikes at the mongoose, and the mongoose flips to its side. Its tail starts kind of doing a propeller. And I see it twist its body, and I look at that and I go, it's doing exactly the same motion that a cat would do. And so different animals have evolved the same sort of writing technique, but for different purposes. For cats, it's because they think they're cleverer than they are and they fall off of stuff a lot. For a mongoose, it's an ability to maneuver when it's hunting its prey.
What interesting findings did you have while you were researching your book?
Quite a lot of surprises. One of which is that the earliest physics paper I found where somebody tried to explain how a cat lands on its feet is from the year 1700. And
wow.
So Isaac Newton was still alive when the first physicists tried to explain how this worked. They were wrong, but it was amazing to just find this. The other thing that I love is that 1894 paper. So Etienne Jules Mary, the photographer, he was a physiologist. He really just was dropping every animal. He was studying the motion of everything. And one of his colleagues said, why don't you drop a cat? So he dropped a cat. He took the photos. I don't think he expected much to come of it, but he took his photos to the French Academy of Sciences, and the French Academy had a huge argument over it because they said, from what we understand of physics, what you've shown us is impossible. And
oh, wow.
And journalists had a field day. I had a friend translate for the book a French journalist who was just mocking the scientists. This fellow was just clearly having a great time going, okay, here are the greatest minds in the world, and they're having this practical, almost a bare knuckle boxing match over how a cat lands on its feet. And so that was just a delight to find that it's an interesting story of how science works, because people had only in the mid 1800s, people really understood that rotational motion has this sort of conservation law that if something twists this way, something else has got to twist the other way to make up for it, what we call conservation of angular momentum. So, but in those early days, people hadn't thought about it too much. So they thought, well, if something's in free fall, it can't just turn over, because it would just be spontaneously rotating. It would violate conservation of angular momentum. But Mary's photographs showed exactly that. They showed a cat being dropped, and then the cat's like, oh, I'm in free fall, and it flips over. And so the French Academy was having this big argument about how this happened. to their credit, by the time they met again, like a week later, they'd all said, "Okay, I think we figured it out. We're good." But it was a little embarrassing. It was a perfect example of the old adage that a little knowledge is a dangerous thing. They thought they understood this problem, but they hadn't really thought about it that much, and they were totally baffled when they were faced with the real world.
The real world, it doesn't fit what my mind thinks. Yes.
There was another version of this that happened with horses running, where all the artist depictions, if you look at the old paintings, when there is a point when horses have all of their feet off of the ground when they're running really fast, and it's when they're outstretched. And you'll see this in all sorts of like gigantic, huge works of art. This type that take up like the side of a house, kind of like big paintings showing horses doing this with it. And then there was this bet that took place where they had to do what was it? It was either Stanford or somebody made this bet.
Leland Stanford.
That's right. And so I guess he won the bet. It was like $1 bet or something, but they had a high-speed photographer go and take pictures of this horse running by. And it turns out there is a point when all of the horse's hooves are off the ground, but it's all tucked up underneath. Yeah, it was sort of the opposite of what they had thought. But
Yep. Not to sound too smug, but I actually talk about that in my book as well.
Oh, nice.
Because that whole idea of high-speed photography was such a game changer for science and naturalism. There were all these things that people took for granted, and then they were shown how it really was. And the artists were actually kind of offended because they had argued for years about the proper way to show a horse galloping. And
well, that just would not look too.
I know, just imagining this grand painting with a horse tucked up underneath itself. It's not as expansive. Yes.
So now as I'm recalling, I think it was Mulridge who did the photography for this. But Leland's bet was that that there was a point when all the hooves were off the ground because he had an art collection with horses doing this like Superman move. And it turned out somebody else argued that never happens. And it turned out he was right. It was just not how he thought he was right.
Well, I mean, it happens when they jump, but
right.
But in a run.
Yeah, no, I'm kidding. High-speed photography is so. Yeah, I was just thinking about how many studies I have brought to this show that are dependent on high-speed photography, and it's how do butterflies move? How do dragonflies stop from falling? That was a really good one.
That was a good one.
How do spiders jump? It's all based on this high-speed photography.
So then if we get even faster, though, and we're trying to figure out how light is operating, isn't there a problem at some point? Because doesn't light kind of just ignore time at some point pretty much and just go, that's your photography can't like chop up.
Can it keep up with it?
Yeah.
In fact, I can say that there has been research done, experiments have been done where people have been able to photograph the motion of light. And they use they're what's called a streak camera. And the idea, I don't know how I can explain this because it sounded better when I wrote it in a page. But so they actually have these images of, so they actually like had a beam of light. They flashed like a light bulb, and their camera was fast enough that you can see the reflection of the light as it travels over a surface. You're actually seeing the motion of light. And it's a bit cheating because in order to get a full image, they have to do like, basically a streak camera can only take a picture of a line of pixels. Because what it does is it separates out the image it sort of keeps track of the time in the vertical direction and takes the picture in the horizontal. And so in order to get like a two-dimensional movie of light moving, they had to do the same experiment over and over again and sort of
You're talking about the inkjet printer, right?
Practically like that.
Perforated sides.
Yeah, but So yeah, but it's a really cool, and I don't think anybody knows what to do with it. It was sort of a proof of principle, like, hey, we can actually make a camera that goes fast enough to see light as it travels. So while again, one of those things that nobody would have thought would have been something you could actually do, and somebody managed to push the technology to get there.
Now that we have the technology to get there, where can that technology take us? Because that's always like in science and technology, that seems to be it always, these ideas build on each other.
Yeah. Well, yeah, I'm not sure. It seems like if you have a camera that can go that fast, I imagine the next natural thing would be to see whether you could design it to measure things that are not light itself anymore, but try to measure reactions like, I don't know, atoms emitting light or absorbing light, try to get images of these sort of quantum level effects that are usually too fast to keep track of. This is just really off the top of my head, because when I first saw that camera fast enough to watch light move, I was like, well, that's really cool, but I have no idea what you're going to do with that.
Sometimes proof of concept is just like, it's a gift in itself.
Yeah. And
science for science sake.
Yeah. And as I've learned, it's whenever you see something like that and you say, I have no idea what you're going to do with that, somebody's going to eventually find something to do with it. So I'm not wise enough to predict it all.
What do you want to do with the invisibility stuff that you're working on?
My own invisibility stuff is partly I'm trying to really understand the physical limits right now, is to see whether we can understand how to make a perfect cloak that works for all colors of light. So part of my research is really along those lines of seeing how we can improve the existing theoretical models to make something that works better. Also, another aspect of invisibility that really intrigues me is the idea of using it for protection instead of just hiding. Because this idea of guiding waves around an object, that should work for any type of wave. It's not restricted to light. So people have actually proposed seismic invisibility cloaks. So I can stick something in the ground around a building, say, so that if there's an earthquake, it can guide some of the energy around the building and might make the difference between the building surviving or not. And in that case, it doesn't have to be a perfect cloak. It just has to do enough to improve the resistance of the building. And there have been some seismic tests of this done. In 2014, some folks took a pile drive. They went out to like a quarry. They dug a bunch of holes in the ground to mimic the structure of an invisibility cloak. They used a pile driver on one end and hammered it down to create like shallow seismic waves, and then measured what vibrations appeared on the other side. We're able to say, by putting this pattern of stuff in the ground, we're able to partially block these minor seismic waves. And so partly my work is still very fundamental, but I always also have an eye towards, it would be really cool to see some of these invisibility and cloaking concepts go towards practical protection applications like that.
Yeah, that's pretty impressive. I was also thinking you could make maybe pretty cool solar panels, or where you could just move you could do a whole surface or a wall with this, and then instead of having all the solar panel you just move it all over to one solar panel that can handle a high intensity beam that's all of the other light of, say, one wall on one side of a building down into a small space, and just have that be the little collection device there. There's all sorts of things you could do if you could control that.
Yeah, and this is sort of, broadly speaking, this is a lot of what people are doing with this idea. The whole idea of invisibility is connected to what people call metamaterials these days. The idea that if you can control the structure of a material on a scale on the order of tens of atoms or hundreds of atoms, then you can create optical properties that you don't find in materials in nature, which we call metamaterials. And with metamaterials, people are doing all sorts of things like, hey, we can make solar panels that collect more light than you would think they should be able to, or make invisibility cloaks, or make a lens, like a camera lens that's flat. Most of our cameras, we have multiple lenses, and you have a really nice camera, you have a huge lens. And so part of the idea is, well, we can smash these lenses down and make a lens that works just as well, but it's just this thin sheet. And people have been trying to do that for years. I don't know if how well they've succeeded or not, but that's sort of still ongoing stuff that people are interested in. Again, breaking the rules of the way things are traditionally done, and saying, we can just try and do that in a completely different way and ignore all the rules and see what happens.
Yeah, and I love the application of things where things are taken from one arena and then are allowed to take different forms in different arenas. So if you're talking about a lens that you can use to collimate light, then maybe there's another a way that a lens like that could be used. A thin, lighter lens would be really useful on telescopes, or on on telescopes, especially if you're going to be launching them into space, right? So,
and speaking of space, then you got radiation diversion devices for habitats on Mars.
Or on Earth. I mean, this is what we need to do, is figure out how to do a protective waveguide to get some of that solar radiation to go around Earth and not just go right into it. Yeah.
Maybe.
We're thinking on bigger scales. I don't know.
Never hurts to think about it, because, in my career, there have been all sorts of things that I've seen that at first glance, I'm like, this just looks ridiculous. This will never work. And then like a few years later, somebody's like, oh, look, here's a practical prototype. And so I've learned to keep my mouth shut at those moments. And if it's not obviously wrong, I'm not going to criticize it.
Let people be creative. People can take things places. Yeah. Is there anything that we've missed talking about? What are you doing next? Are you writing another book? What are you working on? I mean, I know pandemic has kind of affected everybody's lives, but
as it turns out, I'm now writing that history of invisibility physics book that I alluded to earlier. Invisibility also has a longer history than you'd think. I can trace it back. In science, I can trace it back 100 years. In science fiction, I can go back, 200 years. So the book is a bit of, okay, here's what the science fiction authors said, here's what the scientists did, and sort of the interplay between how science fiction authors thought it would work and how it ended up actually working or not working.
Very cool. So we'll definitely have to get you back on to talk about your book when you're done with that. But
so one quick question about invisibility. Can I ask one? Since you brought up the science fiction thing. If I myself was invisible, would I need to take my clothes off to be invisible?
It depends on the type of invisibility you would use. Like, if you were going to have an invisibility cloak that would guide light around you, then you could keep your clothes on. If you were doing some sort of chemical invisibility where you were altering your own body structure to make you invisible, then you'd either have to be able to apply that same process to your clothes or lose them.
Okay.
Yeah, and you'd have to shave your head. That's you'd have to, because that's dead material. It's not going to be affected. All the hair has to go. All of it.
Yeah. And nobody
like the worst green screen ever.
Yeah. And nobody ever explains what happens when you're invisible and you get dirt on you. I mean, you stand in a room for 10 minutes, you're covered in dust.
You're a big dust bunny.
Yeah.
Yeah.
You almost asked my
funny
favorite
shape of dust, like a person.
Yeah. That's weird. That wasn't there before.
Kiki, you almost asked my favorite question at the end there. I think you kind of did, but then maybe you backed off of it, which is there a question we didn't ask that you'd like to answer?
I wish I had a good answer in the form of a question to that question.
Well, then there isn't. Then we covered it all.
The answer's 9 to that question, right? It's 9.
And And this took us back to the inverse problem in our questioning.
Yeah, we'll start with we'll start. Do you have any good answers? And then we'll formulate the questions to go with those later.
Getting panic flashbacks to my PhD defense now.
Same, yes.
But no, I'm good. This has been fun.
Oh, it's been so great. Thank you for joining us. Where can people find you? And and your book that is out right now.
My book is Falling Felines and Fundamental Physics. You can find it through any bookstore or online bookseller. It's from Yale University Press. Gregory J. Gbur is what it's under. Otherwise, I have my blog, which is called Skulls in the Stars, skullsinthestars.com. It's really sort of a hobby blog. So I write about science, history of science, Dungeons and Dragons, book reviews of horror fiction that I find entertaining, whatever catches my mood. And I'm also on Twitter as Dr. Sky Skull. There you'll find me often just making snarky comments and ranting about things. And
ranting viral comments.
Yeah, sounding borderline unstable, though usually it's an act.
I have to say, I have to just admit to our listeners here that yours is one of my favorite Twitters to read.
Thank you.
Yeah, it is amazing. It's very well curated of, absurdity and candor and like outrage and quirk. It's great. I love it.
Well, thank you. I really just try and be honest and also try to be informative and not too grim.
I think that is that's a good balance and hard to take.
Yep.
Thank you so much. I don't want to keep you up too late tonight. It's been wonderful to speak with you and your cats.
Thank you. It's been great talking to all of you live, and hopefully we can do it again sometime.
I would love that. Thank you so much. Have a great night.
You too. Bye.
Take care. Bye. Hey, everyone. Thank you so much for joining us for This Week in Science. That was a fantastic interview. If you loved the interview, we love to bring episodes and interviews and shows like this to you every single week. So you can help us out by heading over to our Patreon page. Go to twis.org, click on that Patreon link, choose your level of support, and help us keep doing this show week in, week out. Your support really is what allows us to do this show. $10 and more a month. It's really not that much. It's like less than a cup of coffee per week. We're asking $10 and more or more per month, and we will say thank you by name at the end of the show. We really can't do this without you. Thank you so much for your support. And now we're going to come on back into the next segment of our show. This is This Week in Science, and it is now time for something we love to call Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
Yeah, Blair.
Oh, I got so excited dancing, I didn't even open my stories. So first, I have a story, kind of a callback from a story a couple weeks ago. I have a new study, this time from, now I lost it already, University of Pennsylvania. Yep, there it is. Okay, I found it. It's because I kept clicking back to the story from several weeks ago from Italy. I was like, no, but that's not it. No, that's not it. It's a proof of concept study, once again, on the efficacy of dogs sniffing out COVID. So this is looking at specifically trained detection of COVID-19. And this was with 96% accuracy. That is better than some COVID tests we are currently using. So the thing that's really interesting about this is that they pulled dogs that had no experience in scent training. They got 8 Labradors and 1 Belgian Malinois. They had never done medical detection work before. And first, they trained them to recognize a distinctive scent, a synthetic substance known as a universal detection compound, a UDC. They used a scent wheel So there were 12 ports. They had different samples in each port, and they rewarded the dog when they found the UDC. So after that, then they started training them to respond to urine samples from SARS-CoV-2 patients and discern the positive from the negative samples. The negative samples had the exact same inactivation treatment beforehand, so they made sure they controlled for anything possible going on with the way that the urine was being processed. And after 3 weeks of training all 9 dogs, they were able to readily identify the COVID positive samples with 96% accuracy. Their sensitivity and their ability to avoid false negatives was a little bit tougher because of a couple things. One is their testing was very strict. So if a dog walked by a port containing a positive sample even once without responding, they couldn't circle back to it. That was a miss. And the other was that they were thrown off by a patient that tested negative, but who had recently recovered from COVID, which totally makes sense that they would carry compounds that maybe would end up being a red herring in that case.
And they have shown that some people, even though they've recovered symptom-wise, do carry a low viral load in their blood for a long time, that they actually, that it wanes. It takes a long time to wane for some people.
Yeah,
so that's
absolutely.
Interesting.
So if
chat room wants sample size. Chat room wants sample size.
Yeah, I said 8 Labrador Retrievers and a Belgian Malinois. So this is 9 dogs. This is again a proof. This is a proof of concept study. Okay, but this is also coming out of the same dog training lab that has previously been used to identify things like ovarian cancer. So this is something that is coming from a pretty fleshed out, pretty robust precedent of dogs being able to sniff out medical issues. And so this is something that is piggybacking on that. And again, if dogs can do this, this could really benefit a lot of the ways that we do COVID testing in the future.
So I'm on the record saying I don't believe any of this.
Yeah, I recall that. That's why I brought this.
Yeah, and it really makes perfect sense. The dog's olfactory sense, their ability to smell is like living in a world with all of the colors and the numbers and the letters and the everything else and the connotations of and the mental connections between what you think about a color and what it feels like and all that all at once in their olfactory. They've got such a high fidelity sense of smell that in nature it is utilized for sniffing out molecules drifting in the wind or latent ones that have been left somewhere on a plant or in the ground so they can track, and repurposing that nature can be anything, can be any molecule.
Well, but also just think about how many animals depend on being able to tell who is sick. How many animals are there in the animal kingdom that we see treat sick individuals differently for whatever reason? Maybe they isolate them. Maybe they give them extra care. Maybe they give them extra food. It depends on the animal and the social connections they have. But this is a very common response. Not to mention, I do think that if we did some studies on humans in this would be a thing. Because have you ever been told that you have sick breath? Because this is something that I have experienced.
Just you.
No, I have experienced on other people. This is a thing, right? Is that this could be totally unrelated, but still there's an opportunity for there to be chemical signaling happening at every level, whether we realize it or not.
Yep.
Yeah, I also, but it was to your point, I was going to follow up with the canines also sniff out the sick because that's who they want to eat.
Yes, also that.
And they're also sniffing out the sick bison because it's going to move a little slower than the rest. They just need to separate it from the herd a bit and then they'll catch it.
Well, not to mention dogs can tell if you're about to have a seizure or if your blood sugar drops. Dogs are very good at these medical things. So there's a whole lot of stuff here. And basically you're going to see more and more these stories propping up. We don't think this is our last coronavirus. So this is a good thing to have in the pocket, even if it doesn't get figured out by the time COVID's over, which it never will.
I just want to make it clear. I'm now on the record as completely believing it.
You flipped. That's great.
A
You come to the
completely
side of
thought
New
science.
evidence.
I
That's
appreciate
great.
that. Okay, and next I would like to move on to Beetle Pee. So this is from University
From
of
people pee to beetle pee. There we go.
So Justin, help me say Copenhagen, Copenhagen,
Copenhagen.
Copenhagen. So
Copenhagen.
it's from University of Copenhagen. Sounds terrible coming out of my mouth. Anyway, this is looking at beetles and how to kill them. Currently, on Earth today, 1 in every 5 animals on our planet is a beetle. There are a lot of beetles on our planet. About 25% of global food production is lost due to insects, mainly beetles. So if we can find a way to control beetle populations, particularly around agriculture, this could be beneficial. The problem is, how do we currently control insects? With insecticides that paralyze an insect's nervous system. The problem with that is it's not very species specific. Nervous systems are similar across insect species, and if you use insecticides, they tend to kill everything, including bees, butterflies, you name it. So it would be beneficial to come up with an insecticide that would just target the pests themselves, without getting into the whole philosophical conversation about what really is a pest, but whatever. We're going to shelve that right now. We're going to recognize that we have the agriculture that we have, and we need to protect it. So, knowing that, researchers from University of Copenhagen,
Copenhagen is fine. Just say Copenhagen.
Yes, okay.
It's fine.
Copenhagen. Wonderful, wonderful Copenhagen. They have discovered which hormones regulate urine formation in the kidneys of beetles. What does that mean? So that means that they can cause beetles to create so much urine that they die of dehydration. Basically, they pee themselves to death. So the inspiration, the inspiration for this study, very interesting. Ancient Egyptians. They knew that if they mixed teeny tiny pebbles into grain stores, that the stones would scratch away the waxy outer layer of beetles on their exoskeletons, which is there to reduce evaporation. And so it would kill beetles due to fluid loss from evaporation because they damaged that waxy layer with the stones. Of course, they would sometimes end up chomping down on some stones in their grain stores, but it was worth it because it wasn't infested with beetles. So knowing that, they wanted to see if they could find a chemical, more targeted way to use this kind of inspiration. So they were able to design a molecule that resembles this hormone. The big kind of question mark here is that in this research, they had to inject the hormone into their bodies so that it would then regulate urine formation. And so this study does demonstrate that the beetle will regulate their kidney function. That first of all, they do it totally different from other insects. And second of all, that they can affect how that kidney function is working through this hormone injection to fatally disrupt the fluid balance in their bodies. So now the next step is going to be to figure out how to get it into the beetles through their exoskeletons by feeding it to them. Somehow they have to figure out the kind of the delivery method to prove kind of this secondary proof of concept to see if it could actually work as a pesticide. But it appears to be very effective at mimicking that hormone and making them just pee forever until they die.
Wow.
I think there are a lot more things to consider. I mean, okay, it's great. They've got this unique protein. Is it really unique? Are we sure if we get this chemical into the environment? Because that's probably how it would have to be released to be taken up by the beetles.
Right.
Could it have any other effects? But this fascinating idea. I mean, it would be like giving somebody a whole bunch of coffee and never letting them drink water.
Yeah.
It's like, well, suddenly diuretic. You've had too much. Yeah.
This is why I'm kind of two minds here. So one, I totally agree with you because yes, this thing mimics this hormone. Yeah. Might it also mimic something else in another species? We don't know. Might this hormone have something to do in another species we haven't found yet? We don't know. So I agree with you there, except for the fact that our current pesticides, we are just spraying out there neuroinhibitors.
Yes.
Just like, let me just destroy the nervous system of all things with exoskeletons out here. So I think in a vacuum, yes, we need to be more measured about this. But recognizing where we live in the current world, maybe it wouldn't be so bad.
Yeah.
I mean, unintended consequences, if we can figure as much out as possible before putting anything in the environment, that's always the better course of action. Although, yeah, we've done so much wrong. Maybe we can start doing things better.
Yeah.
Right?
Yeah. And then speaking of doing things better, I have one more quick story in the answer. It's about snake venom. This is from Clemson University, looking at snake diets and the type of venom that they have in their body. And they found that the number of prey species a snake ate, that did not drive venom complexity, but the types of prey species and how different they were from each other evolutionarily did drive venom complexity. So if a snake eats 20 different mammal species, not very complex. But if it eats a centipede, a frog, a bird, and a mammal, very complex venom. So they have to have an arsenal that is more diverse to be able to eat all of these different things, which could help us create better antivenoms. One, which, we have a whole database of antivenoms in the United States, worldwide. It's pretty amazing, but it could be better. And so knowing the most we possibly can about snake venom will save lives. Definitely. But secondarily, venom is also often used to create medicine. And so the more we know about venom, there's a potential for kind of unlocked drug capabilities that we don't even know about yet. So for example, snake venom derived drugs treat heart disease, high blood pressure, blood clots. Makes sense. A lot of venoms are anticoagulants, right? So they can take a lot of that information and help human medicine. So not only is this helping us understand snakes better just evolutionarily and for the interest of the study of snakes, but it's also helping us save lives from snake bites and potentially unlock some interests for human medicine.
That's fantastic. That sounds like it's win-win, win-win, right?
Knowing more about snakes is a win-win.
Yeah.
Also because they're cool.
They are cool. They're so cool. Do you appreciate a bit of honey with your tea? A little honey in the cocktail you might be drinking? A little honey on your peanut butter and banana toast?
Ooh. Oh. Banana. Out of here.
Right. Well, okay. So, bananas could be a little bit radioactive, and it's because of the potassium they take up. Well, a lot of plants actually, they take up potassium, and they can actually get fooled by radiocesium, which is radioactive cesium, and they can take up this radiocesium in place of potassium, and it gets incorporated into their tissues and into their pollens. And a researcher and his students just found that flowering plants across the country probably are still incorporating radiocesium from the nuclear tests that we did back in the 1950s and 1960s. And that honey samples brought by the students when they went away for spring break back to the campus, at least half of them, a little more than half of them, were radioactive and had radiocesium in the honey. It's not a lot of radiocesium.
It's not going to kill anybody.
It's not dangerous. It's well below dangerous radioactive levels. But what this implies is that, there's all sorts of foods that we eat, all sorts of things in our environment that are sticking around, and that decades ago, over 50 years ago, that we did nuclear bomb testing, and the Russians did nuclear bomb testing, that it got the radiation ended up in the atmosphere, it ended up in the soil, and that is still circulating. And that some organisms can concentrate that radioactivity, and then it continues to cycle and concentrate.
So future scientists will find our bodies and just see them full of
plastic
isotopes and plastic. Yes.
Wait,
like, what were they doing?
Who are these people? Yeah.
How did they even live?
Yeah. But it's an important part of figuring out how our ecosystems work to understand stuff like this. So may not have been too surprising to a lot of people, but, you know, a lot of people eat their honey because it's healthy, and now maybe going to go, oh no, radioactive honey. But it's not that bad. It's just maybe just a little bit of radioactivity.
Yeah.
It's fine. It's totally fine. And then I wanted to dive into some fun stories about T-Rexes, because for whatever reason, this last week, there were about 3 stories about T-Rexes that they were so fascinating. So first off, T-Rexes were slow, and imagery in movies of these storming T-Rexes able to run at 30 miles an hour. That's really, really not likely. They were very, very heavy, and most likely T-Rexes were very slow, only walking, maybe moving at about 3 miles per hour, 5 kilometers per hour.
Well, if they were scavengers, then what are they running for? They're giant.
Exactly.
Nothing's hunting them.
So were they hunting or not? Another study found that T-Rexes I love this, in a quarry in Utah that is called the Rainbows and Unicorns Quarry. It is they've found a number of fossils. Many of them seem to be a group of T-Rex individuals that died together. And their hypothesis, it was the result of either some kind of biological poisoning by a microorganism or fire or flooding. The preferred hypothesis at this point is that they died due to a flood altogether. But the fact that their bones are all found together adds more evidence to a growing body of fossil evidence that T-Rexes were not solitary hunters, that they were social group animals, and maybe they hunted in packs or in a cooperative manner, or scavenged in a cooperative manner.
Think about turkey vultures. There's always a bunch of them because they're following the scent, right?
Right, if they're scavengers. So maybe the other hypothesis there isn't that they scavenged together, but maybe they followed the scent, slowly moved their way to the dead thing, and then a group of T-Rexes fought each other over dead things.
Or a carcass fell into some quicksand, and then they all just kept following it in.
They all kept falling in, which is, highly likely. And finally, the last study was another piece of work estimating, and this was in Nature, I believe, but another piece of work estimating the number of tyrannosaurs that had potentially lived on Earth. They estimated it to be some 2.5 billion dinosaurs over a period of 2.5 million years, mind you. So this is a long period of time. But that there could have been at any one time about 20,000 adult T-Rex individuals between 68 million and 65.5 million years ago. Yeah, this was in Science.
So just to let you know, there are about 3,900 tigers left on our planet, to give you an idea here.
And if we think of T-Rex as a major part of the ecosystem, how big they were, how many other dinosaurs did there have to be for 20,000 T-Rexes to be crawling around at one time? So when you start thinking of ecosystem interactions and number of individuals to support individuals, then you start realizing how many dinosaurs there were. We need to find more rainbow and unicorn quarries.
Mm-hmm. Yeah, and I also looked up, there's about 39,000 giraffes left on our planet and around between 12 and 15,000 bison, American bison. So more than there are bison. Just picture T-Rexes roaming around.
Well,
and I'm going to picture them in big groups.
Yeah.
Thousands of T-Rexes with their little arms. Yeah, and this is interesting also because of the fact that this study specified only 20,000 adult T-Rexes because they only looked at that form. And now we know that juvenile and adults had different body forms and they survived eating different kinds of foods. So how many juvenile T-Rexes were there? Lots more. Oh, yeah. Fascinating view of that, was it Cretaceous period of North America's history. This isn't like all over the world either. It's like North America. Anyway, my final story for the night, I want to talk about a glance, a gaze. What happens when I look into your eyes? What happens when you catch somebody looking out of the corner of their eye and then looking at you? What does it make you do? How does it make you feel?
Do I know them or not? I feel like it's very dependent on if I know them or not.
Regardless of whether you know them, other people's gazes capture our attention. And a new study that's out this week from the University of Geneva finds that when people make eye contact with other people, it solicits that attention. Not only is attention solicited, that cognitive process distorts temporal perception. So you actually overestimate, or you actually underestimate the amount of time that was spent being consciously aware of that gaze, because your brain is working on it. And so time is dilated when people look at you and you look at them. You have mental time dilation because of people's eyes looking at you. So the researchers had different situations and measured what was going on in people's cognitive functioning, and they measured their perception of time, had them tell the researchers how long this thing took or that thing took as they were looking at these faces. And they had these faces look at the subjects, either just a face staring and not moving, a moving face where the eyes came to look at the subject, just the eyes moving to look at the subject, or inanimate objects. And they found that the eyes moving and the head moving are a very important part of this time dilation. So if there is just like a mannequin with its eyes straight, and your brain recognizes that as a human face, nothing happens, because that face, you might have passed its field of vision, but that face is not actively looking at you. It's just gazing into the distance. But the faces that moved and the eyes that moved, whether or not it was just the eyes moving or the whole head and eyes moving, those elicited time dilation in the brain. So this also gets at this question of, how masks are potentially affecting our ability to, to communicate with people, they're not affecting our gaze. And even if your face, your mask, your lower face is covered, your eyes are still going to be able to solicit attention in other people's brains and start cognitive processes that lead to connection.
So human gaze is a whole thing with dogs too.
Oh, yeah.
Yeah, so dogs are really good at following human gaze, but for example, wolves are not great at it.
I thought this is happening again. I thought it was the opposite.
Okay, I'll double check, but I'm pretty sure that is
I
the
thought
situation.
that the wolves were actually better because the whole point of that study was the fact that we thought this has been this sort of shared history training up of dogs watching us and us watching dogs over the millennia. And then it turns out wolves are just better at it than dogs.
So my recollection is that this was their proof that dogs and wolves have a much farther back evolutionary convergence. And so this was actually an indication that it was something that dogs developed completely separately and in their own timeline, apart from other canines.
I remember the result of that being completely the opposite. So we have to go look at this. We have to go find this out.
Well, I found the study. It's from 2019. So,
Wait. So I had it backward. I'm going to go find my study.
I'm reading it right now.
Okay. We'll do it in the after show. We'll
talk about it in the after show. Fine. I'll put this in the show notes for later. But anyway, gaze is something that we study with humans in relation to other animals, is my point. So gaze is an important social signal, not just human to human. That was the whole reason I brought it up.
Yeah. It's important. It's important across animal species. And it is interesting also for different species animal interactions. The animals we interact with, whether they're other human animals or dog animals or cat animals. How do the gazes interact? But amazing that just catching a gaze can affect your sense of time.
Mm-hmm.
Mental time dilation with a look. Hey, Justin.
Hi. Oh, yeah.
Things?
Yes. Okay, so when you think of a creature that travels really well, one that has spanned the globe, a super disperser that goes everywhere, one creature that probably doesn't come to mind as one of those you'd find everywhere, anywhere kind of creatures is the tarantula. Tarantulas are very much like hobbits from Tolkien's Middle-earth. They're not fond of big adventures. They prefer to keep to their burrows, only occasionally leaving to find food or mates. Unlike hobbits, though, tarantulas are absolutely everywhere. So how did this, the 6 out of 7 continents have tarantulas on them? So how does a big hairy spider that doesn't like to leave the burrow, that has usually a very tightly controlled area within which it operates, how does that creature end up spreading across the entire planet? An international team of researchers, including Carnegie Mellon's University's Saoirse Foley, set out to sort of figure out how this came about. They did basically like a genomic 23andMe type of thing for the tarantulas, trying to figure out where their genes come from. They've published this in online. Peer J is where it's been published. You want to go look it up there. So they used transcriptomics to build a genetic tree of spiders, and then time calibrated their tree with fossil data. And despite the fact that tarantula fossils are extremely rare, they were able to sort of backtrack tarantula emergence from places. So they found out that they considered America's about 120 million years ago, Cretaceous period. So aside from there being 20,000 T-Rexes, there were also plenty of tarantulas in North America at the time. And South America as well, India, Australia, all these places that they reached, they did so mostly a long time ago. They found a couple of lineages that colonized Asia about 20 million years apart from each other. But they actually made it across the boundaries between Australia and Asian islands, where many other species sort of have a hard stop from exploring into. And basically what it came down to, why the tarantula is so successful, such a global trotter, is that it's really good with niches. They're just really good at finding a little spot in the food web and owning it without having to force anything out or compete too much with anything. In fact, one of there's been the species that inhabited, I think, Asia at the time, were both a forest dwelling, like an arboreal dwelling, like a tree dwelling version, and a burrowing version that didn't interact with each other, that didn't intermix, because they both had their little niche, and they just kept to it, little burrows, kept to the little hobbit shire, didn't explore, didn't like rabble rousing. Didn't like troublemakers, and just by doing that, they're really good. In fact, some of the continental conquests that they made were because the continents moved away from each other or crashed into each other. That's how they got from India to Asia. Is the continent crashed into the other continent, and everybody's like, okay, I guess it's one more continent.
We'll just take that. I'll do that.
Yeah.
Whether by burrow, by tree, by air. Yeah, just as long as there's no flying tarantulas next.
Yeah, but the looks of it, it's just by not being, even when being invasive, not being too off-putting when you get there. Yeah. Not taking up much of a footprint, even though you've got 8 legs. Shift work. My final story today.
Let's hear this bad news.
So here's the thing. It's often been maligned as bad for your health. But will this new deeper dive study make us rethink benefits associated with a flexible work schedule? No, there's nothing. No. There's nothing good. According to research from University of Waterloo, shift work is just plain bad for human health. Causes all sorts of health-related issues that affect our defense against disease and infection, because when you mess with the circadian clock, you disrupt the sleep-wake schedule and feeding patterns, and these are the things that actually are part of your immune system's rhythm as well. Interestingly, though, it appears as though it affects men worse than women. Men have a larger negative reaction than women, according to the study.
We've talked before about hormones being involved, and I think it's just such an interesting question as to what would make it work that way. Mm-hmm. Why? What does melatonin or any of the circadian clocks, how does that affect the immune system differently in the different sexes with different levels of sex hormones? And
so next I need to figure out if there is anything that actually helps, because there's melatonin you can buy sun lamps, you can do all sorts, you can eat chocolate. We talked about that on the show once. So I want scientific studies that actually shows what can help offset that a little bit.
One would be day job. Day job will help.
Not everyone can be a day job.
I know. Well, everyone can work towards a day job.
All
even
I'm
if
saying
it's a little
is
less money.
When you get hurt in the middle of the night and you have to go to the ER, you're very happy that there are people working at night in the ER.
Yes. So
if this is affecting inflammation.
Are we just talking about Brian now?
Yeah, now we're just talking about Brian. Yeah. So we start understanding exactly how shift work affects everything, and then we can start really targeting the responses and the treatments to it. But if what we are looking at is a disruption of the inflammatory physiological response, then how can you ameliorate inflammation? Right? So I think the next step would be, regardless, as a shift worker, what would be the things that you could do in your life to decrease inflammation. Anti-inflammatory diet. I mean, you don't necessarily want to take NSAIDs every single day because that's not good for your liver and kidney. But what else can you do to make just reduce the possibility of inflammation?
Yeah, so part of it too is I guess there is natural times like you're not supposed to get an infection right before bed because your body is better prepared to fight off infection the next morning.
That's why you always feel really bad right before bedtime. Like, I don't feel good anymore. Your body's like, no, go to bed.
But there's natural cycles of our immune system sort of beefing up and shutting down, and these cycles that we go through. And when you mess with circadian rhythms and you don't let your body have those sort of natural cyclical processes that take place on a daily basis, it's overproducing or underproducing or just giving up on you completely. Just forget it.
Don't let it give up.
Yeah.
Never give up. Never give in.
And then in the case of the men, I guess the immune system tends to overreact, leading to potential higher rates of sepsis.
Oh yeah, so the men are so strong that they're feeling extra bad.
Well, I mean, there might be some sort of like ancestral biological thing about men getting more infections.
Yeah,
I don't know.
This is man flu. This is just more man flu stuff.
This is more. Oh, that's.
But I mean, similar that like COVID is an inflammatory disease, and men are affected worse more often. So it's there is precedent, and man flu is a real thing.
Yes.
Yeah, I was going to say, Blair, this is probably the first time I'd ever heard you say that men don't overreact to things that affect women. It's just
men aren't overreacting. They actually feel bad.
Yeah.
When they feel bad, they feel bad. Come on.
Yeah. Have a little bit of
as someone said in the chat room, men do have a higher stress level than women. So it's tough to be a man.
Well, it is, because we have to, not only do we have to deal with all the stresses of, society and all this, we also have to deal with women.
You should just show yourself out right now, Justin.
Yeah. Bye, Justin. See you never.
Bye.
Tune in next week to find out who the third host is.
It's Rachel, isn't it? It's going to be Rachel.
Oh, have we made it to the end of the show?
Yeah.
So,
did we do it? We got through all the stories. This was a fun show.
Might not have gotten there soon enough. Minute early, I would have been okay.
Thank you for helping to host this show. Thank you, everyone, for listening. And shout outs to the people who really helped this show. Fada, thank you for help with social media and the show notes. Extra work this weekend with the crossover show. Very, very appreciated that you spent time on that.
Awesome.
Gord, thank you for manning the chat room. Identity 4, thank you for recording the show. Again, being there this weekend. It's fantastic. Everyone who did show up live on Saturday to see our crossover show with DTNS, thank you so much for being there. Love the fact that you joined us it was a really fun show. It was really great. And again, if you are a Patreon sponsor, then you'll be getting that show in your podcast feed over there as well when I put that in. Rachel, thank you for your assistance. Very much. Thank you. Thank you to our Patreon sponsors. Thank you to Carl Kornfeld, Jen Myronik, Melanie Stegman, Akramsta, Karen Tazi, Woody MS, Andre Bissett, Chris Wozniak, Dave Bunn, Vegard Chefstad, Hal Snyder, Donathan Stiles, a.k.a. Don Stylo, John Scioli, Guillaume, John Lee, Ali Coffin, Gorov, Sharma, Shubrew, Darwin Hannon, Darnold Mundus, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Josh Fury, Shauna Nina Lamb, John McKee, Greg Riley, Mark Hessflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Kevin Railsback, Flying Out, Christopher Dreyer, Mark Mazzaros, Artyom, Greg Briggs, John Atwood, Robert Coburn, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Matt Sutter, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Sarah Chavez, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, EO, Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Stanton, Paul Disney, Patrick Pecoraro, Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you for all of your support on Patreon. And if you are interested in supporting us on Patreon, head over to twist.org and click on that Patreon button. And on next week's show,
we will be back Wednesday, 8 p.m. Pacific time, 5 a.m. Central European time, broadcasting live from our YouTube, Facebook channels and from twist.org/live.
Hey, do you want to listen to us as a podcast? Maybe catch up on your science while you do some dishes? Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twist.org, and you can sign up for our newsletter.
You can also contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, somewhere in that subject line, or your email will be spam filtered under an invisibility cloak. We'll never see it.
You'll never find it again.
Yeah.
You can also hit us up on the Twitter, where we are at Twist Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you'd like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. 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 And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 This week in 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 science.